The effects of antioxidant enzymes on sleep deprivation in Drosophila melanogaster. Millions of people in the United States suffer from sleep disorders. Sleep is increasingly recognized as important to public health, with sleep insufficiency linked to motor vehicle crashes, industrial disasters, and medical and other occupational errors. Unintentionally falling asleep, nodding off while driving, and having difficulty performing daily tasks because of sleepiness all may contribute to these hazardous outcomes. People experiencing sleep insufficiency are also more likely to suffer from chronic diseases such as hypertension, diabetes, depression, and obesity, as well as from cancer, increased mortality, and reduced quality of life. Sleep insufficiency may be caused by broad-scale societal factors such as round-the-clock access to technology and work schedules, but sleep disorders such as insomnia and obstructive sleep apnea also play an important role. An estimated 50 to 70 million U.S. adults have sleep or wakefulness disorders. Figure 1 shows the self-reported sleep-related difficulties among adults over 20 years old, reported to the Center of Disease Control and Prevention from 2005 to 2008. Concentration on things and remembering things being the leading cause for sleep loss. A study of the effects of partial and total sleep deprivation on driving performance was conducted jointly by the Federal Highway Administration, Human Factors Laboratory, and the Walter Reed Army Institute of Research. It examined the effects of progressive sleep deprivation on stimulated driving performance in the laboratory to assess the rate of accidents and changes in driving performance resulting from sleepiness. Figure 2 shows that within 4 days of sleep disturbances and shortened sleep, there was an increase from an average of no car crashes in the stimulus to an average of 8 car crashes a day. Figure 3 shows the circadian rhythm clock in humans. The circadian rhythm dips and rises at different times of the day, so adults' strongest sleep drive generally occurs between 2 and 4 a.m. and in the afternoon between 1 and 3 p.m., although there is some variation from person to person. The sleepiness we experience during these circadian dips will be less intense if we have had sufficient sleep and more intense if we have had sleep deprivation. The circadian rhythm also causes us to feel more alert at certain points of the day, even if we have been awake for hours, and our sleep and wake restorative process would otherwise make us feel more sleepy. Figure 4 shows a comparison between a Drosophila and a mammalian circadian clock. The common fruit fly, Drosophila melanogaster, is a well-studied and highly tractable genetic model organism for understanding molecular mechanisms of human diseases. Many basic biological, physiological, and neurological properties are conserved between mammals and Drosophila, and nearly 75% of human disease causes genes are believed to have a functional homologue in the fly. In a study done by Everson, Latch, and Hogg, they tested antioxidant defense responses to sleep loss and sleep recovery. Sleep deprivation in the animal model produces a condition that eventually becomes highly lethal, lacks specific localization, and is reversible with sleep, implying mediation by a biochemical abnormality. Metabolic and immunological consequences of sleep deprivation point to a high potential for antioxidant imbalance. The objective for their study, therefore, was to study glutathione content in the liver, heart, and lung because glutathione is considered a major free radical scavenger that reflects the degree to which a tissue has been oxidatively challenged. They also investigated major enzymatic antioxidant catalase, an enzyme in which that catalases the reduction of hydrogen peroxide. Catalase activity and glutathione content, which normally are tightly regulated, were both decreased in the liver by 23 to 36% and by 5 to 10 days of sleep deprivation. Sleep levels are associated with impaired health in other animal models and oxidative stress-associated diseases. The decreases were accompanied by markers of generalized cell injury and absences in responses by the other enzymatic antioxidants under the study. In a study by Cove, Evans, Hendricks, and Senga, they experimented with different ages of the flies and how age correlates with sleep cycles. To identify changes that occur over the course of a lifetime, they monitored the sleep and wake behavior of individual flies in activity tubes continuously from a few hours after eclosion until their death. Reports that flies undergo an age-associated breakdown of sleep and wake cycles reminisce of that seen in humans. Because an increase in oxidative stress disrupts sleep and wake cycles in the manner similar to that of an aging, they propose that the accumulation of oxidative damage with age, at least in part, accounts for the breakdown of sleep and wake cycles. Although there were some differences in sleep patterns between young male and female flies, sleep became more evenly distributed over the 24-hour period with age for both male and females. The results concluded that the sleep cycles were most constant in male and female flies between the age of 2 weeks old to 2 months old. The figure shows an average sleep cycle of a fly 10 days old in the black line and an average sleep cycle of flies over 40 days old represented by the gray line. As flies age, their sleep cycles become fragmented and male and female cycles start to change. For this study, the model organism used will be Drosophila melanogaster. There will be 4 groups. The first one will be the control. For the control, the flies are fed regular Drosophila medium and left alone during the night without being exposed to any light so they can sleep normally. For the second group, the flies will be fed regular Drosophila medium and subject to sleep deprivation for one entire day. The third group is fed Drosophila medium with glutathione mixed in with it and will also be sleep deprived for one whole day. The last group will be fed Drosophila medium with catalase mixed in with it and will also be sleep deprived for one whole day. The flies that are being sleep deprived will be left under a flashing light to keep them awake throughout the entire night, while the control flies will be separated and put somewhere in the dark. After each day, the death count of the flies will be counted and longevity will be checked immediately after the sleep deprivation day. The number of deaths and longevity will be analyzed by an ANOVA followed by a post-hoc Chauvet test. The purpose of this experiment is to see if antioxidant enzymes such as glutathione and catalase will have an effect in reducing the effects of sleep deprivation. The alternate hypothesis for this study is that the antioxidant enzymes will reduce the effects of sleep deprivation. The budget for this experiment is a total of $116, including the Drosophila melanogaster, the medium, the vials, the glutathione, catalase, and the focusing LED flashlight.